Hello friends, welcome to Sam Biology class. Today we shall discuss about dosage compensation and lion hypothesis. Before that, I request all of my friends, those have not subscribed my YouTube channel, please subscribe. In species with XX and XY system of sex determination, for example in human being, both male and female possess 22 pairs of autosomes. But regarding sex chromosomes, there is a difference between male and female. Females possess a pair of X chromosomes, but males possess one X and one Y chromosome. So there is a difference in the number of X chromosomes possessed by male and female. This difference presents a special problem in development. Because females have 2 copies of every X-linked genes and male have only 1 copy, the amount of proteins produced by X-linked genes would differ in male and female. Because of this, female would produce twice as much gene product as that is produced by the male. This difference could be highly detrimental because protein concentration play a very important role in development. Animals overcome this problem through dosage compensation. Dosage compensation is a mechanism which equalizes the amount of proteins produced by X-linked genes in male and female. In Drosophila, dosage compensation is achieved by doubling of the activities of the genes that are located on the single X chromosome in male. In the worm Sinorhabditis elegans, dosage compensation is achieved by reducing the activities of the genes that are located on both X chromosomes to half. Placental mammals use yet another mechanism of dosage compensation in which 1 out of 2 X chromosomes is inactivated, thereby equalizing the amount of proteins produced by X-linked genes in male and female. Now, what is lion hypothesis? In 1949, Murray-Barr observed condensed darkly staining bodies in the nuclei of female cats. These darkly staining structures were named as Barr bodies. Barr bodies. In 1961, Mary Lyon proposed that Barr bodies are the inactive X chromosomes. Barr bodies are the inactive X chromosomes. Her hypothesis has become known as Lyon hypothesis. Mary Lyon proposed that in cells with 2 X chromosome, one is inactivated. Which X chromosome is inactivated is random. In cells with more than 2 X chromosomes, all X chromosomes except one are inactivated in order to achieve dosage compensation. Because of this X inactivation, females are functionally hemizygous at the cellular level for the X-linked genes. In females that are heterozygous at the X-linked locus, 50% of the cells will express one allele and remaining 50% will express the other alleles. So both the alleles will be expressed within the same individual but not within the same cell. Functional hemizygosity means Cells in the female are not identical with respect to the expression of the genes on X chromosomes. Thus, females are considered as mosaics for X-linked genes. Dosage compensation takes place due to random inactivation of X chromosomes that occur rarely in the development. In human beings, inactivation of X chromosome takes place early during the first few weeks of development. After a particular X chromosome has become inactive, it remains inactive permanently not within the same cell but also in the other cells that have descended from it. Because of this, neighboring cells have the same X chromosome inactivated, and this produces a patchy pattern for the expression of X-linked genes in heterozygous female. According to Lion hypothesis, presence of variable number of X chromosomes should not affect the phenotype. This is because any X chromosome in excess of 1 must be inactivated. But individuals with Turner syndrome differ from normal female, and individuals with Klinefelter syndrome differ from normal male. Now the question is, how do these abnormal conditions arise in the face of dosage compensation? These disorders such as Turner syndrome and Klinefelter syndrome arise because some X-linked genes escape inactivation. The mechanism of inactivation of X chromosome is more complex than it was originally suggested. Study of individual X-linked genes reveal that about 75% of the genes located on X chromosome are permanently inactivated. 15% completely escape inactivation. That means the genes are able to produce twice as much product as they do in male, and remaining 15% of the X-linked genes are expressed in some females but not in others. The reason for this variation in the expression of X-linked gene is yet to be known. So friends, this is all about dosage compensation and Lion hypothesis. For more update, please subscribe my YouTube channel, Sam Biology Class.